your respiratory tract, your urothelium or your urinary tract, your reproductive tract, your oral cavity, all of the lining of your mouth, your esophagus, all the way down to your anus, your vagina, your reproductive tract, all of that is lined by epithelia. Okay, the lining of your glands, the pancreas, the salivary glands, all of that is epithelial tissue. And so it is designed for the movement of substances, specifically water, okay, secretion and absorption. Now, there are two membranes across these cells. There's an apical membrane and a basolateral membrane. So if you look at this cell, so think about this as being the inside of your gastrointestinal tract. So food is moving through this lumen. It's going from your mouth down to your anus, and it's going to be in contact with the apical surface of this cell. Right? At the basal surface or the basolateral surface, that's going to be coming in contact with the interstitial fluid and the underlying blood vessels of this tissue. Okay, so the purpose here is to have the nutrients and water from the food we eat move through the epithelial cell and deposit those nutrients and water into the interstitial fluid and the blood vessels which are underlying the epithelia. So there are also tight junctions. Some of the features are tight junctions. And these tight junctions prevent leakage of substances in between the cells. So let's say you have sodium or chloride that is moving through these cells. You don't want any substance leaking in between the sides here. And so that's the purpose of these tight junctions. We're going to speak about them in much more detail in the integumentary system. There's also the apical membrane, which we mentioned earlier, and the basal or basolateral membrane. Now, there are certain features on the basolateral membrane that are not present on the apical membrane and vice versa. So let's start by looking at what's present at the basolateral surface. So on the basal membrane or the basolateral membrane, there is the presence of the sodium-potassium pump. We spoke about this pump before. And it's going to be bringing potassium in, specifically 2 potassium in and 3 sodium out, okay? What type of transport is this? What type of transport is this an example of? This is active transport, right? Primary active transport, which requires ATP, all right? Also present on the basolateral membrane are potassium channels, and these are ion channels. So these are going to be moving potassium according to its gradient. These are leak channels, as well as a glucose carrier, right? A glucose carrier. What type of transport is that glucose carrier an example of? What type of transport is that glucose carrier an example of? So we spoke about carrier proteins, and these are examples of facilitated diffusion, right? So we're using some sort of mechanism, specifically a carrier protein, to transport glucose out of the cell here. Okay. On the apical surface, there are sodium channels, right? Leak sodium channels. There are sodium-linked glucose pumps as well. So the sodium leak channels are basically moving sodium into the cell freely, passively, according to the gradient, whereas the sodium-linked glucose pump is using the energy that was harnessed from the movement of sodium to power glucose moving against its concentration gradient. Okay, what type of example is that? What type of transport? What type of transport is the sodium-linked glucose pump? Perfect, thank you. Secondary active transport. So we're using the energy that's harnessed from this ion moving down its concentration gradient, and we can move glucose against its concentration gradient. Okay. So let's look at the example of the movement of sodium or sodium transport here. So because of the sodium-potassium pump on the basolateral membrane, we're kicking sodium out, we're bringing potassium in. Now we spoke about the pump leak system. The pump leak system is where ions that are pumped out of the cell or into the cell, ions that are pumped can then leak back according to their gradient, right? Leak back down their gradient. So you pump free sodium out of the cell, sodium is going to go around the other end and leak right back into the cell. And that is what maintains a steady concentration of sodium inside and outside the cell. Same thing with potassium. You pump 2 potassium into the cell, and potassium is going to leak right back out. Okay, so that combination of pump and leak, pump leak, it's what keeps the concentration gradients, the concentration of the ion stable. Okay, so sodium then leaks back into the apical surface, and in doing so, it's going to generate energy that we can use to bring glucose against its own concentration gradient. Okay. And we already described that secondary active transport, where sodium is being brought in down its own gradient, and that energy is being harnessed to transport glucose into the cell as well. Right? So when glucose comes into the cell, it's then going to be moved across the basolateral membrane via facilitated diffusion. So we're bringing glucose in against its concentration gradient, and because we're using energy from this movement of sodium, this is called secondary active transport. When glucose enters the cell, it's then going to be transported via this carrier, right? And because this is a carrier, this is facilitated diffusion. So glucose is then going to be transported out the basolateral membrane and into the blood, right? So this is how glucose and other molecules from the food we eat get from the lumen, where we bring it into the GI tract, out into the blood. Now, in this scenario, where is the concentration of glucose higher? I want you to think about that for a little bit. Where would the concentration of glucose be higher given what we just said? Where is the concentration of glucose higher? Is it higher inside the cell or outside the cell? Come on, take a guess. Inside the cell, right? That is correct, right? And we say inside the cell because glucose is moving against its concentration gradient, right? And because it's moving against its concentration gradient, it's moving from an area of low concentration to high concentration. So that right away tells us that there must be more glucose inside the cell than outside the cell for it to be moving against its concentration gradient here. Can everyone see that? Can everyone see that? Okay, let's move along. All right, so we spoke about the movement of some ions across an epithelial cell, and now we're going to speak about water movement across epithelial cells. So the way that water moves across these cells is very unique. All right, the cell has to secrete certain solutes first. It's going to pump certain solutes out into the extracellular fluid, right? So this cell will pump solutes, sodium and chloride, out into this interstitial fluid here. And that generates a gradient. So because we have more solutes here than we do in the lumen, water wants to flow from the lumen down into the interstitial fluid. Okay, so we very smartly created a gradient by pumping sodium and chloride out into the interstitial fluid. We created a gradient where we have a salty or a more solute-rich environment here than we have up here. And so we have more water here. Water's at a higher concentration here. Water's at a lower concentration here. And so water naturally moves according to its concentration gradient, right, via osmosis, through this cell right out into the interstitial fluid. Okay, so we essentially created a water gradient using the excess solutes here as an osmotic pressure or an osmotic pull to pull water from the lumen. Okay. Does everyone understand that? So water movement here is happening secondary to the solute concentration or secondary to solute movement, right? Now let's talk about cystic fibrosis. Does anyone know what cystic fibrosis is? Has anyone ever heard of cystic fibrosis? Yeah, it's kind of common. It's a disease that has many symptoms, mostly respiratory symptoms, and it's classified by thick respiratory mucus, lots of lung infections, upper respiratory infections, and this is because of a defect in the chloride channel of the respiratory airway and other areas. So as we just mentioned, in order for water to move, right, and this is all types of water, water being absorbed, water being secreted, water is a constituent of mucus that we produce, so water has to be secreted in order to make mucus more liquidy. Water is secreted as a part of sperm, as a part of other gastric juices and things like that. And so having a defect in this chloride channel, we cannot pump chloride out of the cell to make this area more salty or have a higher osmolarity, okay? And because we cannot do that, we do not have a gradient where water wants to move. And so what we end up with is a thicker, more sluggish mucus, which clogs the respiratory airway and is responsible for many, many respiratory infections, lung infections, that thick mucus production, and so on and so forth. These patients often also have infertility because the sperm, the quality of the sperm is, or the semen rather, is too thick. And so it's not easily transported to the egg. And so lots of symptoms, all owing to the defective chloride channel, which really impedes the flow of water because we cannot create that solute gradient and we cannot have movement of water. Right, and that is cystic fibrosis. Now there's lots of therapy. I think there's gene therapy that they're working on to see that they can find a cure for this, but as of right now, there's no cure for cystic fibrosis. All right, and the last thing we're going to talk about is transcytosis. Transcytosis. And this is the movement of macromolecules across the cell. Okay, this is going to involve two very important processes that we're going to look at later on, endocytosis and exocytosis. Think about endocytosis and exocytosis as a Pac-Man that is swallowing a certain substance, moving through the cell and spitting it out. So endocytosis would be that engulfing or that swallowing, and exocytosis would be that spitting out at the basolateral surface. We're going to see this process very, very extensively. When you look at the neuromuscular junction, the neurotransmitters are packaged in this way, and we're going to see this endocytosis and exocytosis a lot. Okay, so at the luminal surface, you engulf this particle or this substance, this macromolecule. You move it through the cell packaged in this vesicle. Now this vesicle is sort of bitten off from the cell membrane, so to speak. So it really is a part of the cell membrane. So what is that made of? What is this vesicle really made of? What is the cell membrane made of? Phospholipids. So the phospholipid bilayer is sort of pinched off. It engulfs this molecule. It moves all the way through the cell. And then when it gets to the basolateral surface, it'll spit that molecule out, and the phospholipids will then align themselves to be a continuous part of the membrane here. Okay? And that is endocytosis and exocytosis.